introduce Ailene MacPherson from Simon Fraser University, who will tell us about allele surfing, hybridization, and the evolution of life history traits. Take it away, Ailene.
So first of all, thank you guys for working me in fairly last minute. So I appreciate the opportunity to get to share with you some of these stories about range expansion that I've been working on. So the two topics I'm going to tell you about today don't flow together particularly well, but I'm putting them both in just to give you a flavor of the kind of questions that I have been thinking about. So, so the first is a story about hybridization, and it's the story about the consequences of range expansion, or what happens after range expansion. And then the second one is a story about complex traits evolving during range expansion. So the first story was motivated by this biological system, which is one of many, many systems I could have picked. But these Pacific warblers at the end of at the maximum of the last ice age were isolated in little glacial refugia on the Pacific coast, and then as the glaciers receded, they expanded across the landscape and eventually came into secondary contact in a couple of places in the Pacific Northwest. So there's many reasons that we might want to study secondary contact hybrid zones. And theory about secondary contact hybrid zones is very old, but here's one classic model by none other than Nick Barton called the tension zone model. So this model of secondary contact hybridization follows this kind of basic structure where you start with two populations that are isolated by some geographical barrier. And then you remove that geographical barrier, and then the two populations integress with one another and form, I don't know why this looks a little weird here, but they form this stable cline, and it's a sigmoidal shaped cline in allele frequencies. And one of the things that we're going to focus on is measuring the width of this cline, which is simply the inverse of the slope at the center of the hybrid zone. All right. So what would we expect this cline to look like? Well, it depends on what kind of allele that we're looking at. So if we're looking at a neutral locus, then that cline will flatten out and will result in a 50-50 allele frequency in the long term. If there is one favored allele, that allele will sweep across the whole hybrid zone and will fix across the range. And then if there's a genetic incompatibility, you'll end up with a stable sigmoidal cline in the allele frequency across the hybrid zone. So there are several different types of incompatibilities we could think about, for example, under dominance. But what I'm going to focus on today is a Bateson-Dobzhansky-Muller incompatibility. So in case you're not familiar with this, the idea is that you have a big A, big B homozygotes are fixed in the ancestral population, and then after vicariance, one of the populations fixes for the little b allele and the other one fixes for the little a allele. When they come into secondary contact, the little a and the little b have never been found in combination with each other before, so hybrids that are formed with little a, little b genotypes can be disadvantageous. So this is a form of negative epistasis. All right. So this tension zone model doesn't really represent what's going on in this warbler system very well because it ignores expansion. So it doesn't account for the fact that the parental populations had to expand across the ranges before coming into secondary contact. So the goal of my co-authors and I on this work was to try to understand, to propose a new model of secondary contact hybridization that involves this parental population range expansion and understand the consequences of this range expansion on hybridization. So why might we think that range expansion would be important? Well, as we're all really familiar with in this room, range expansion can lead to gene surfing, or what I call allele surfing, where one allele can fix on the range edge, even if it is deleterious. So all types of alleles will surf, beneficial alleles, neutral alleles, and deleterious ones, but we might be particularly interested in recessive deleterious alleles because of how common they are in the genome. So you might imagine that this gray recessive deleterious allele fixes on the range edge due to increased drift. But what happens when we have secondary contact hybridization is that we actually have two scenarios of allele surfing going on. And in some loci in the genome, the recessive allele will fix in one parental population, and the corresponding dominant beneficial allele will fix in the other population. What's really interesting then is when you hybridize between these two, if the hybrids, the F1 hybrids, will have the dominant beneficial genotype or phenotype, and it will mask this recessive deleterious allele. So what this can do is it can result in heterosis at this locus. So higher hybrid fitness than the average of the parents. All right, so that potential for heterosis is what motivated this project. So we see extensive amounts of variation in heterosis in crop plants. Some crop plants exhibit extensive heterosis, other ones almost none at all. And it's been an enduring mystery about why this is. And so could the potential history of range expansion actually explain the variation in heterosis that we see? Second of all, if these mass deleterious loci are linked to these genetic incompatibilities, could the heterosis that results help these genetic incompatibilities intergress across the range? And this would result in a breakdown of the reproductive barrier and prevent speciation. And then the last part of this is, as you guys may be familiar with, Stephen Peischel has shown that there can be a significant impact of recombination on the pattern of allele surfing and the extent of range expansion. So how does this whole story depend on recombination? All right. So here is the individual base model simulation setup that we used. So we start by simulating the two parental populations in allopatry. This is pretty boring, but essentially we let them reach eco-evolutionary equilibrium. We then let the two populations start expanding across the landscape. During this process of expansion, we create these artificial F1 hybrid test experiments represented by these squares so that we can look at how heterosis accumulates during the process of range expansion. And then we let introgression proceed and we look at the patterns of introgression. And we tried 5 different scenarios of recombination. We tried a scenario of free recombination. We tried a scenario where you have limited recombination, in that recombination is distributed very unevenly across the genome. In particular, you essentially have 4 different chromosomes. So this is very similar to what Peischel did. And what they call recombination is actually much more similar to segregation, where you have independently recombining regions, but within those regions, you do not have recombination. So to complement the segregation scenario, we tried 3 different recombination scenarios, one where you have a low rate of recombination that's at the same average as the chromosomal one, but is uniform across the genome. And then 2, where the pattern of recombination across the chromosome reflects what we see on average in animals and plants. All right. So, and all except for the high recombination rate scenario have the same average recombination rate. Only thing that's changing is the distribution of recombination rates. So what do we see? Well, the first thing we want to see is just do we see expansion load? So are we seeing this pattern of recessive deleterious allele surfing? And yes, we do see an accumulation of recessive deleterious alleles that results in a low fitness on the range edges of both parental populations. And this can be shown by this increase in expansion load. Okay. So what happens if we look at the F1 hybrids over the course of expansion? Well, we see heterosis accumulate. So early on, heterosis accumulates really quickly, and this isn't surprising because you have this standing genetic variation that's leading to lots of accumulation of heterosis, and then it slows down. There are several other determinants of heterosis. The strength of selection against those recessive deleterious loci will change the extent of heterosis. It's a non-linear pattern, so it will not increase the amount of heterosis forever and ever, but for at least weak amounts of deleterious recessive selection, it will increase heterosis. The dashed lines here represent partially recessive mutations instead of fully recessive, and this just gives you a fraction of the heterosis. Heterosis also depends on the amount of recombination, whether or not we have BDMIs. So if you have a BDMI, a dominant BDMI, which means that all hybrids experience that incompatibility, this will just reduce the amount of heterosis that we see. All right, so that's what we see during range expansion. What happens after range expansion? Well, we can see that same pattern of heterosis that we saw during the expansion playing out on a larger scale once integration occurs. So that heterosis leads to this triangle of high fitness at the point of secondary contact. So what was once just one population of F1 hybrids with high fitness spreads out across the range. And one of the cool things about this is it leads to a different shape of decline in allele frequency at these recessive deleterious loci. So if you remember back to the tension zone model, we imagined that the decline in allele frequency would be sigmoidal shaped. It is no longer sigmoidal shaped. It is what I'm going to call secant shaped, and that is because allele surfing, the deleterious alleles can be in really low frequency in the parental core, and then they surf to high frequency, and this leads to this secant shape. All right. But even like the tension zone model, over time, those recessive deleterious alleles are eventually swept out of the population, and you're fixed for the beneficial allele across the range. So what happens at the incompatibility loci? Well, first of all, the incompatibilities can have a dramatic effect on fitness. So in this plot here, I'm showing the reduction in fitness due to the incompatibilities, and especially where there's lots of hybrids, we can see that there's a strong reduction in fitness. But the width of this BDMI integration is given by the width of this colored region is wider than we would expect in the absence of any recessive deleterious loci. So this is measured by the Klein width. And so as the strength of selection on those recessive deleterious alleles increases, the amount of linked selection on the BDMIs increases, and this helps them integress. So parental range expansion can help break down barriers to gene flow. And the whole story depends on the pattern of recombination that we have. So not only does it depend on the overall rate of recombination, but the more unevenly recombination is distributed across the chromosome, the more these BDMIs are able to hitchhike with these masked recessive deleterious alleles, and the more the speciation will be prevented. All right. So that kind of brings me to the conclusions of the first little section of this, which is that range expansion can have really important consequences on heterosis and what we observe when we cross F1 hybrids. And then it can actually prevent speciation from occurring by helping the barriers to gene flow like BDMIs introgress. I just wanted to note that while we focused on hybridization and secondary contact here, that this result is not really specific to this. And another cool implication of this would be on hybrid invasions. So if you have an invasive species that can hybridize with the local population, then the fact that the invasive species may have expanded across the range could result in a burst of high fitness once it hybridizes with the native population. and promote invasion. All right, so this is the end of that story. But while we were simulating these, we noticed something weird about allele surfing and the accumulation of expansion load, which the next project is going to dig into. So our understanding of allele surfing and the accumulation of expansion load is kind of limited in its scope currently to understanding the accumulation of density independent selection. So alleles where the strength of selection doesn't depend on the population density. However, we know that many traits actually experience much more complex forms of natural selection, and we can see 3 different forms of selection simply by writing out this life cycle. So this is just your classic logistic growth model. And we could imagine that selection acts either on the intrinsic growth rate, the carrying capacity, or the viability of individuals. And then if we calculate the strength of selection acting on each one of those traits as we would measure it in a single population, so we can see that the selection on intrinsic growth rate is negative density dependent. So selection is strongest when populations are small and weakest when populations are large. Here you have stronger selection at the bottom of this axis because selection is deleterious, and so the more negative, the more deleterious it is. So this is strong selection, weak selection. In contrast, selection on competitivity or carrying capacity shows the opposite pattern, where selection is weakest when populations are small and strongest when populations are large. And then in contrast, viability, which is what we use for the vast majority, or the analogous thing to viability for the vast majority of allele surfing simulations, has a constant selective pressure over the course of over population size. So since population density varies over range expansion, we might imagine that these traits will evolve very differently on the range edge and accumulate deleterious alleles at very different rates. So what we did is we simulated range expansion in a linear stepping stone model, and then we have a complex genome consisting of 300 loci, 100 loci underlying each one of these traits. These loci are a very small effect, as you might imagine. And then we measure the mean allele frequency for each one of these traits for the loci underlying each one of these traits over time. All right. So what do we see? Well, first, some predictions. So this is the model of selection in a single population. Once again, when populations are large, all 3 traits have the same selective pressures, but when populations are small, carrying capacity is under weak selection and growth rate, intrinsic growth rate, is under strong selection. So if we map these population sizes over here on this x-axis to the realized population size during range expansion, we can just essentially flip this plot around by 180 degrees so that selection gets weaker and weaker and weaker over time on carrying capacity over the course of expansion, and stronger on intrinsic growth rate, and then remains the same on viability. And you might notice these light dashed lines. Not only are these traits density dependent, have density dependent selection, but they also experience epistasis. So these lines indicate different levels of epistasis on these traits. So what do we see in the simulations? Well, we see that carrying capacity or competitive ability accumulates the most deleterious mutations, whereas growth rate, intrinsic growth rate, accumulates the fewest deleterious mutations. So we can't directly interpret this mean allele frequency in terms of selection. There's lots of reasons that they may accumulate different rates or different amounts, but the rate at which they accumulate deleterious mutations should be proportional to the selection coefficient. So if we look at the rate at which these mean allele frequencies are changing, that pattern should look fairly similar to what we see in the selection coefficient. And indeed it does. So they accumulate overall, the rate of accumulation of deleterious mutation decreases over time, just due to the fact that you're running out of standing genetic variation and you're having to accumulate mutations from alleles from new mutation. However, relative to the blue line, we see a pattern very similar to what we'd expect given the selection coefficient, where the carrying capacity is under significantly less selection than intrinsic growth rate. So what this all means is that allele surfing is not going to happen in all traits, and the amount of expansion load will depend on the nature of density-dependent selection. So negative or positive density-dependent selection will accumulate more or less expansion load and will show different patterns of allele surfing. And this has implications for how we measure expansion load. So this is an ongoing project, so I don't have the figure to show you, unfortunately. But essentially, depending on the population size in which you estimate expansion load, you will get vastly different answers. So you have to be aware of the population size in which you're measuring fitness when you talk about expansion load. All right, so that's the end of my 2 stories. Here's the summary of the major points. And once again, thank you for making the time for me to talk.